Morning, everybody. Okay, I'm going to get started. Again, office hours today, 9 to 10. Crowded, but please come anyway. We can always stuff more people in the room. And tomorrow, 9 to 10, where there will be fewer people there. So if you can come on Tuesday, 9 to 10, it's a good time to come to get things taken care of. So today I'm going to finish talking about transport across membranes and move on to something new and different, hopefully in about 15 or 20 minutes. At the end of the hour, I was talking about proteins last time, and I showed the behavior when you transport a molecule with a protein. It was this curve I put on the board, and it's important for you to understand this curve. So this is the concentration of material being transported, and this is the rate of transport. And the behavior of this curve is important. The curve goes up, and then it levels off at some optimum or higher level of transport. The reaction is said to saturate, which means, because it's protein mediated, you have a limited number of protein molecules in the membrane which are transporting whatever it is you're transporting, and eventually they're all occupied, they're all busy. So increasing the concentration of a molecule to transport doesn't increase the rate any. Very, very fundamental behavior that we'll see again when I talk about enzymes. If you look at figure, I hope everybody has this book and looks at the figures when I refer to them. Figure 73 summarizes the different kind of transport proteins that exist in membranes. And I'm going to briefly say a few comments about how these things move across the membrane, how say, polar or charged molecules, because several people have asked me, a couple people have sent me emails, how do you really get these molecules which are charged across the hydrophobic bilayer? It's a good question. Okay, so you have carrier proteins off to the left. These are proteins that have the capacity to pick up a molecule on one side of the membrane and move it through the protein and deposit it on the other side of the membrane. These proteins, as do other kinds of transport proteins, will change conformation. Conformation is the structure of the protein. Remember that from one of my first lectures. So what happens is when a molecule picks up, say, a glucose molecule on one side of the membrane, that changes the shape of the protein enough so that the glucose can be transported across the membrane and deposited on the other side. Channels are a little different. They're shown in the middle portion of that figure. Channels are essentially proteins forming holes in the membrane for generally small molecules like ions to go through. And there was a picture on previous page 28, of the potassium channel, where you've got all these helices in the membrane, and they form basically a hole in the membrane. And the hole size dictates what can come through and what cannot come through. Another sort of channel type protein, which is interesting, is, I'll just mention it because I think it's particularly interesting, is a protein called aquaporin. This is the structure of aquaporin. And you can see it's got a lot of helices. It's a membrane protein. The function of aquaporin is to transport water across the membrane. Fairly unusual reaction. There are some cells that take up water very, very quickly, and people couldn't figure out how they did this, and finally someone realized there is a pore, a hole, a protein-mediated hole in the membrane that actually functions in moving water across the membrane. And finally, the other protein that exists, other types of transport proteins, are those that are involved in active transport. And the unique feature of these is they can move molecules against the thermodynamic gradient. They can move from high to low, not high to low. you've got a high concentration inside a cell, they can continue to move a molecule into the cell where there is already a high concentration. These proteins all require energy, and the energy source that is generally used is this molecule ATP, which we'll talk about in more detail later. But you should understand that there is an energy requirement. Now let me just say make a comment about how do these things move across a membrane. Firstly, in the case of some molecules, the membrane is fluid, and some of them can actually move through the phospholipid bilayer because of the fluidity properties of the membrane. But what's more common is that I said when you have these helical regions in a membrane, they're very hydrophobic, and that's true. But every now and then you find a protein that has in one of its helical regions a non-hydrophobic, perhaps a polar amino acid or charged amino acid. For example, in this aquaporin molecule, there is one asparagine residue that's in the membrane region. In a protein we'll talk about later in terms of ATP synthesis in mitochondrial membranes, there's a section of an enzyme that makes ATP, very hydrophobic region because it's moving protons across the membrane. There's a carboxyl group. So there are specific, maybe one amino acid. If you see an amino acid like a charged amino acid in that hydrophobic bilayer region, it has undoubtedly got to do with moving that molecule across the membrane. The other thing you have, of course, are carbonyl groups, which are in peptide bonds. And, for example, in the water aquaporin, they are believed to play an important role in moving molecules across the membrane. The problem with all of this is we don't really know. And it's hard to work with these proteins because when you take them out of a membrane to isolate them, you can't have a transport process, right? You don't have an inside and an outside. So firstly, they're very difficult to get out of membranes. And secondly, they're very difficult to study if and when they are removed from membranes. You can get a crystal structure, but that doesn't tell you that, well, it starts to tell you something about the mechanism. So it's a very, very difficult, these are very difficult systems to work with, and progress has been slow, but not impossible. There's another way one can characterize transport proteins, and that's based on what they transport, or the type of molecule they transport. There's a figure of this as well. Figure 74 shows there are three types of carrier proteins. Carrier proteins is okay. One which simply transports one molecule at a time. That's a uniport. The second is a symport, where it transfers two molecules, but they have to be transported at the same time. There is a proton glucose transporter, and you need a glucose molecule and you need a proton, and they are transported together. And then there's an antiport, which is one molecule in one direction, another molecule in the other direction. This classic example of this, I mentioned already, is the sodium-potassium transport system, which I'll just say a few words about it in a minute. And it's important for you to realize that uniport, symport, and antiports are used for active and passive transport. Of course, the active transport also requires energy. I've stressed that before. So there's an energy requirement for active transport, but not for passive transport, because those are going with the concentration gradient. I've said the classic example of an active transport system is shown in figure 75 schematically. There's a good figure in your book of the sodium-potassium transport enzyme. These enzymes go by various names. They are called pumps, the ATP-dependent enzyme. They're called ATPases because they cleave ATP. So you have to be aware the pump the sodium-potassium pump and the sodium-potassium ATPase is the same protein. And these proteins clearly undergo conformational changes. For example, when sodium binds to the protein, it changes the basic conformation of the protein so that potassium binding is weak. When sodium is released, then potassium binding can be stronger. So there is a lot of sort of basic biochemistry going on. The enzyme would be a lot easier to understand if it was 2 sodiums going in one direction and 2 potassiums going in the other direction, but it isn't. I've put in the numbers. It is 3 sodium ions transported in one direction and 2 potassium ions transported in the other direction. Take a look at the picture in the book. I believe that this enzyme, Mike, you can confirm, it uses 50% or some large amount of the ATP that's made in a cell. A third, okay, 33% of the ATP generated in a cell is used to drive this transport process. So it's a pretty important one, and you'll be hearing more about it later. Okay, finally, what I want to just point out very briefly again is there are situations where the cell can take in large sort of chunks of material, and this is shown in figure 76. Okay, figure 76 to the left shows you the exterior of a cell, and the membrane, the plasma membrane, has this fluidity about it, and it can basically invaginate, form a vesicle where stuff is trapped inside that vesicle, and then that vesicle will bud off and will generally move to the lysosome where the material in there will be degraded. Similarly, you have an exocytosis scheme where material inside the cell, this is basically how secretion is done. We've talked about secretion before, where material inside the cell coming from the Golgi complex is in a vesicle. That vesicle will fuse with the internal membrane, the plasma membrane, I mean, and be released to the outside. So this is two sort of macro transport processes. Maybe I should also mention that there is, of course, a mechanism, or mechanisms, probably plural, for how proteins cross membranes. This is what I talked about in terms of targeting. It's not well understood. As I've said, I think several times, you should appreciate by now, after this discussion of biological membranes, how difficult it would be to get a large, high molecular weight protein across a membrane. And there's clearly a receptor, and somehow that protein has to go across, say, the mitochondrial outer membrane or the chloroplast outer membrane, things like that. So this is also a very, very difficult area to study. If you look at page 32, that is, again, a series of study questions, because I finished with membranes and cell structure. And again, these are for your use. And if you're having problems or you don't understand something, come to office hours and ask me about it. Or there are also GSIs in 2084. I want to make that point too. Okay, big turn in direction now. What we're going to be talking about from this moment on is cellular metabolism. And I will bring next time my schemes, my big metabolism maps, so you can see how complicated metabolism actually is. Metabolism is the chemical process that is occurring in cells. It's the synthesis and degradation of all the molecules we've talked about. Remember, it's important to remember that I sort of stress molecules as they're formed. Biological molecules in a cell don't live forever. They are broken down, they're degraded, and the component parts of them, amino acids, nucleotides, fatty acids, are reused to synthesize new macromolecules. So there is this constant synthesis and degradation. This is what's shown in this particular figure. We have food, that's a general term. sugars, fats, all the things we eat. These are heterotrophic cells, obviously. And these foodstuffs are being converted to carbon dioxide and water. And we will spend a fair amount of time understanding how that process occurs. When this is occurring, there is energy available. I had written over here energy released. I don't want to. You don't want to get the feeling that the energy is just lost. What has to happen in a cell is the energy that's made available in this degradative reaction has to be trapped by the cell and used for synthesis. And that will be the focus of the last 4 or 5 lectures. There are various terms that are used to describe these two processes, and you'll hear them. They're interchangeable. Catabolic or exergonic reactions are the degradative reactions. They release energy, or they make energy available. Anabolic or endergonic reactions are the synthetic reactions which require energy. It takes energy to build a protein, for example. Okay, now I used to talk a lot at this point about thermodynamics. Don't ask me why. Free energy, first law, second law, all of those things. And I don't do that anymore, so don't worry about it. It's in the book, actually. The book does talk about the first and second law of thermodynamics. I don't do it because those thermodynamic principles are not really directly applicable to biological systems. They deal with the energetics of the universe, and I'm not interested in the energetics of the universe. What I am interested in, and what biochemists have been interested in, is a function, a thermodynamic function, which is known as the free energy function, delta G0. The free energy function is the amount of energy for any specific reaction that is made available for work. That's in a degradative process. When you're breaking down molecules, there is energy available, and the free energy function will give you some estimate of how much energy is available. And it's related to the heat content of the molecules and this fascinating term that is philosophers have worried about and scientists have worried about, the entropy term, which is some measure of loss in the process. So every process, there are losses. There is no 100% efficient energy conversion. And the entropy factor gives some measure of loss. The 0 on the delta G, delta H, and delta S. This is a point of some confusion. Sorry about it, but blame the thermodynamics people. Delta G0 is measured under what are called standard conditions. Under standard conditions. These turn out not to be very biological. For example, the pH may be 1, or the concentrations are generally 1 molar, something like that. They're very arbitrary conditions that don't relate directly to biological systems. But they do allow delta G's, delta G0s for various different chemical reactions to be compared to each other if everybody measures them under standard conditions. You will also see delta G. In fact, this is what is in Russell. And he uses delta G wrong. He's really talking about delta G0, but he has delta G. Delta G are non-standard conditions. But it doesn't matter. This is not terribly important. But it is, the difference is important because what we would like to know is the free energy change for a reaction under cellular conditions, not at pH 1 and 1 molar concentrations. Those don't make any sense. Okay, here's a reaction that we will be studying in some detail, and it includes a number. There aren't many numbers that I want you to be responsible for. This is one of them. This is what happens when glucose, C6H12O6, plus oxygen are converted to carbon dioxide and water. The free energy change for this reaction is minus 686 kilocalories. That means 686 kilocalories are either being relieved, they're available. I'll try to use the term are available for every mole of glucose that is being actually oxidized, in this case, converted to CO2 and water. This 686, where does it come from? It comes from the bond strength of the molecules that are starting products versus the bond strength and structure of the molecules that are the products of the reaction. Now, you have no idea, unless you have read future chapters or had some bioenergetics before, what that number means. Is that a bit? Is it a lot? Is it a little? And the significance of the number doesn't mean anything at this stage of the game. The fact that it is a negative 686, minus 686, has been interpreted or has been described as this reaction being a, I hope I have room, spontaneous reaction. You'll see this a lot in the literature. Spontaneous. Can you see that? That is, reactions that have a negative free energy change are spontaneous reactions, and that's wrong. It's not a good term, and I will stress in a minute why it's a bad term actually. What is really happening in this particular reaction is there's energy being released. It doesn't necessarily occur spontaneously, but there is energy available when these compounds are formed from these. In contrast, if you take CO2 and water and you make glucose and oxygen, the reverse reaction of what I had up there, that reaction requires 686 kilocalories of energy because I'm using the same materials, right? There and there versus there and there. So there has to be an input of 686 kilocalories of energy to make glucose. There is a release of 686 kilocalories of energy in the oxidation of glucose. This turns out to be what we call respiration. And this turns out to be photosynthesis. And the energy required for photosynthesis comes from sunlight, right? Sunlight drives all of these processes. You can see there's clearly a relationship between these two processes. One of them is making glucose, photosynthesis. One of them is degrading glucose, respiration. So there is some cycle that goes on in terms of sugars and oxygen. One of the interesting features of the free energy function is if I look at various systems that carry out this reaction. For example, the reaction will occur in a human liver cell. Delta G0 is going to be 686 because it's the same reaction. If I go to a bacterium like E. coli, very simple or relatively simple organism, the free energy change is going to be plus 686. If I take what we used to do, and we did this once in physical chemistry way back when, you take a bomb calorimeter. Yeah, you still do that, you see. You know, and you close it down and you pump oxygen in there, and then it will burn the glucose just the way we burn the glucose, and the free energy change is 686 kilocalories. So the delta G0 is independent of the pathway of transformation. I mean, E. coli and a liver cell may be degrading glucose in different ways, but the free energy change associated with the reaction is identical. Everybody okay with that? Yeah. Oh yeah. I'm sorry. It was here. It was right. Yeah, no. This is, thank you. This should be negative, 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 right? Anybody else want to bring that point up? Yeah, okay. Now, we come to my favorite demonstration in the course. I do this every year. I've been doing this for 20 years, okay? It never fails. That's why it's a great experiment. This is actually, this is not glucose, it's sugar, it's sucrose, but it's the same as glucose. So it doesn't change the outcome of the experiment. Glucose is on the table. I guess that's enough. I had 2 packages. Okay, there's glucose, right? There's oxygen in the room. Everybody accept the fact there's oxygen in the room. Yeah? Why isn't anything happening? But there, okay, 686 kilocalories of energy are released. Well, you know, what's the problem? There's a problem. Thermodynamics has a problem. Thermodynamics gives you no information on how fast the reaction occurs. Kinetics. There's kinetics and there's energetics. So the speed of the reaction is not considered in thermodynamic considerations. Generally, when I've done this, I've poured my glucose or sucrose on the table. It's been on Friday, and I've always prayed that they would not clean the tables off. But the guys in the back here are really efficient, and they take the glucose away. That glucose will be there for a long time, is my point. And that's because we have no information on how fast these reactions take. Now, I think it's fair to say that we are some of us are rapidly moving organisms, okay? Some of us are slow at times. But if you look at cellular metabolism, the reactions are fairly rapid. And we cannot wait for the eons that it takes for that glucose to be oxidized by molecular oxygen. So what do we have in our system that makes the whole thing work? We have catalysts. Where can I go? I'll go up here. Catalysts. What are catalysts? Catalysts are compounds that increase the rate of reactions. Without being consumed. So they work over and over and over. During the reaction, we'll say. Now, when I was taking biochemistry, we could make a very simple statement about biological catalysts. We would say then that all biological catalysts are proteins, i.e. enzymes. And one of the reasons I really love biology, this was as firm as anything we had in biology in the 60s. All biological catalysts were enzymes, proteins. And one of the beauties of biology is you can take the firmest idea that we have, the basic foundation of modern biology, and somebody can go and do a stupid experiment and destroy it. Except it isn't a stupid experiment. Well, what happened in the 70s, I suppose, into the 80s, 2 scientists in particular, Tom Cech and Sidney Altman, discovered that there are catalysts, which are RNA molecules. RNA can act as a catalyst. Yeah. So this statement, all biological catalysts are enzymes, is wrong. We now say there are catalysts in biological cells. They can be proteins or they can be RNA. These RNA catalysts are known as ribozymes. And you will hear about them in the second portion of the cell, of the class, okay? I don't talk about ribozymes. I will talk the rest of this lecture and the Wednesday lecture on the structure and the function of enzymes. And if I can remember to bring my metabolic chart in, that I want to stress, that stresses the fact that all of this biology, all of this metabolism that's going on in the cell involves enzymes. There are lots of enzymes, and enzymes work to increase the rates of reactions. How do they do this? What's the magic of an enzyme? Somebody mentioned before something about activation energy, and that's true. If you look at any chemical reaction, there is always an activation energy barrier that it has to go through before it can occur. And the classical figure is the one in figure 77, which looks like this. Try to draw it the same. You have the course of a reaction. Versus the free energy change for the reaction. So the reactants start off with a certain amount of free energy, and the products for a reaction that has a negative delta G are less than that of the starting material, i.e. 686 kilocalories. And there is an activation energy that you come through. This input of energy, I don't know if they call it, well, we can just call it E sub A, activation energy. A certain amount of energy has to be put into the system to sort of get it over the hump, and then it's all downhill. The difference between this level and this level represents the delta G0 for this particular reaction. So that's an uncatalyzed reaction. What an enzyme does is it lowers the activation energy for this reaction. So if I had a different color, I would use it. I don't want to use blue. Blue's no good. Okay, well, I don't have it done. Basically, you go from here, you have a lower activation energy for that reaction. The activation energy has been decreased by some large factor, and clearly this reaction is going to go faster than that reaction. So enzymes, their general mode of action is to decrease the activation energy for a reaction. Okay. How do they do this? Well, enzymes are proteins. And you understand something about the structure of the protein. When a protein folds up into its tertiary conformation, its native conformation, the folding of the protein yields something that is known as an active site. So if you look at figure 78, that blob is an enzyme. You've got an enzyme and it looks like this. Okay, this active site is the region where the protein, the enzyme, will interact with a molecule known as the substrate. Substrate will bind to the active site of the enzyme, and at that active site, the substrate will be converted to product. The active site of an enzyme comes about because of the folding of the protein. Remember, you make a protein, it can basically be in a long linear sequence, but it isn't in a long linear sequence. It has a three-dimensional structure, and that three-dimensional structure is its native conformation, and there is a region in every protein, every enzyme, where the enzyme will bind with the substrate, and the substrate will be converted to product. For example, since I have sucrose on the table, sucrose is a substrate, and sucrose can be converted into glucose plus fructose. The enzyme that does this, its name is sucrase. That's the enzyme that converts sucrose into glucose and fructose. You're going to work with an enzyme in lab. When is that? A couple weeks. Next week, yeah, good. Timing is good. You're going to work with an enzyme in lab that is known as amylase, and its substrate is starch, or you could say amylose and amylopectin. So the term ACE assigned to something in this particular game means it's an enzyme. Enzymes are highly specific. An enzyme such as sucrase that works with sucrose may not work with maltose, which is another disaccharide, may not, well, I would go further, I would say will not work with maltose, will not work with lactose. There is a specificity that is involved in substrate enzyme interactions. So that's why we have so many enzymes, because they are highly specific. Let me just say they're specific for certain types of compounds. I've put in a table about names of enzymes because the names are, there was a convention many years ago to try to name enzymes in a reasonable way, and because the names are common names, they just, you know, when you discovered an enzyme, you gave it a name, and that pretty much has failed. People use the historical names for enzymes, and it's a little hard to read table 79, but you should get some feel for the kinds of, the classes of enzymes there are and the general names of them and what they do. For example, nucleases, the second thing on figure 79, nucleases are enzymes that degrade nucleic acids. Proteases are enzymes that degrade proteins. Synthases are things that synthesize something. So there's some you know, rationale to it, but then when you get down to kinases, what does that mean? Where did that come from? I don't know where it came from, but kinases are enzymes that attach phosphate groups to things. So once we get into ATP and things like that, we will be talking more about kinases. We will be talking a lot about what are enzymes known as oxidoreductases or dehydrogenases. These are enzymes involved in various aspects of respiration and photosynthesis, and they're going to be a major focus of my talk the next couple of weeks. And then I've mentioned already ATPases. These are enzymes that hydrolyze, take off a terminal phosphate of ATP and do various things. So this is some background in naming of enzymes. Figure 80 is an interesting figure. I like figure 80 because it shows you a protein to the left, and it's got various amino acid side chains indicated, you know, in little circles, and the amino acid side chains are all over the protein. The protein folds so that all of these components that were, say, out here and here and here, they all end up in this pocket. Okay, they're there, like that. And that becomes the active site of an enzyme. And what is shown to the right of that figure, the schematic, is a substrate bound in an active site in a crevice in an enzyme. And the only point I want, this is not anything you're not going to sit down and memorize this figure. But I want to show you the kinds of forces that are involved in binding a substrate to the active site of an enzyme. So you can see There are hydrogen bonds. There may be some ionic bonds. There may be some hydrophobic interactions. These are generally weak bonds that bind a substrate to its active site because these molecules have to come in, they have to be manipulated, and they have to leave. The products have to leave so that the enzyme can work again. One question that generally is asked by people is, does the protein, I said it's not consumed, is the enzyme altered? Does it change in the carrying out the particular reaction? And the answer, yes, it does change shape subtly. And I'll talk again a little bit about this maybe in a minute or maybe next time. Subtle changes occur, but it always has to go back to its original starting conformation so that it can interact with another substrate. That's a very unique interaction, substrate-enzyme interaction. So you have enzyme plus substrate forming an enzyme-substrate complex, and then the substrate is converted to products, so you have enzyme plus products. If the substrate stays around too long, this enzyme isn't going to work very well. If the products stay around too long, this enzyme isn't going to work too well. If you can look at figure 81, figure 81 gives you some numbers which are turnover numbers. Turnover numbers are numbers of molecules converted per, I think, enzyme molecule per second, something like that. And it shows you a series of enzymes, carbonic anhydrase, I'll talk about again next time, an interesting enzyme, 600,000 molecules of product are formed per second. That's pretty good enzyme. You can see there are other numbers that are relatively high, 1,000, 2,000. Then there are some enzymes where they look like they're kind of slow, sluggish. Lysozyme, 0.5. It's still catalyzing a reaction, but it's not a very efficient enzyme. Tryptophan synthase, 2 molecules versus 600,000. So these numbers vary all over the map depending on the enzyme, depending on the pathway it is involved with. And the fact that a number is small doesn't mean this is an enzyme that is not so important, for example. It has nothing to do with the importance. When the enzyme binds the substrate at its active site, there are things that happen to the substrate molecule. For example, the reason the substrate can be converted into products is the binding at the active site puts a certain amount of strain on various bonds within the substrate molecule so that they may be cleaved or converted to something else. And this simply does not happen at any appreciable rate in solution, i.e. that glucose, sucrose is still sitting there. So I think you can also see that if I have a reaction which is a little more complicated than this is the classical reaction that we always talk about. We have A plus B, okay, go into the active site of an enzyme and are converted to, guess what, C plus D, okay? The point is you can have two substrates have to interact with each other and they are converted to two products. Now in solution, the probability that substrate A and substrate B are going to meet and combine with each other and get converted to C plus D, I'd say are low, whereas the active site can hold one substrate in one certain position and the second substrate in a nearby position and facilitate the conversion of A plus B into C plus D. So when you have multiple substrates, I think you can see maybe a little better how an active site can really facilitate reactions going on. Okay, I don't know. Okay, I'll talk about one more thing, and then it'll be barely enough. The generalized mechanism for enzyme structure is shown. Two generalized mechanisms are shown in Figure 82. Again, in the old days, enzymes were viewed like locks. There was a lock and key mechanism. You know, you have a key. I have a key somewhere in my pocket. I have a key. This key just fits right into a lock. I turn the key, the lock opens. Everything is preformed. The active site sits there. The substrate comes in. It fits into the grooves and crevices, and it can be converted into product. There's no flexibility in the enzyme. That model was discarded in the sort of 50s and 60s by Dan Koshland was the main manipulator of the old model. Dan Koshland is a biochemist who used to be at Brookhaven, came out to Berkeley, and was a very famous scientist, but also a very good friend of the university in that the building my lab and office is in is Koshland Hall, named after Dan Koshland. A very good protein biochemist. And he came up with an idea which is called induced fit. So we got rid of this idea of a rigid, stable protein, which doesn't change shape at all, and we came up with the idea shown at the bottom of figure 82, where you have the active sites sort of there. They are there in a sort of more nebulous way. As the substrate starts to interact with the protein, there are changes in the shape. And these are not big changes. These are subtle changes in the shape of the protein to facilitate the binding of the substrate. And it shows there distorted bonds and things happen like that. And then the product can be formed, and then the enzyme can go back to its original structure so it can react with a new substrate molecule. So the presence of the substrate is inducing changes in the active site of the enzyme. So this is a more flexible. It sees enzymes and these enzymes as flexible in the context of being able to react in subtle ways to the presence of a particular substrate. Whereas the old idea, lock and key, was, you know, everything was geared to just moving in, don't change the structure, everything was preformed, and it's clear that proteins can undergo subtle conformation changes. I talked about them in relation to transport. It's certainly true in enzyme action, the mechanism of enzyme action, that there are subtle changes in the structure of the protein which facilitate substrate to product conversions. Okay, I'll stop there. Good place to stop, and we'll continue next time. And we'll see if the glucose is still there.